Good morning. Today I'd like to talk about an article that looked at sperm DNA fragmentation and chromatin decondensation and phospholipase C efficacy in infertile patients. This is the article in question. It came out in Current Issues of Molecular Biology. Sukayani Yazil and other authors from a Morocco clinic have presented a paper linking the relationship between sperm DNA fragmentation and phospholipase C functionality in cases of total failed fertilization. So the theory. This is an interesting open access paper from Moroccan authors that explores the relationship between sperm DNA fragmentation and the concentration of phospholipase C zeta in the sperm. Phospholipase C is the soluble factor in the sperm head that activates the waves of calcium oscillations that activates the oocyte and initiates fertilization. These waves in the new zygote influence the initial rate of cleavage in human blastocysts and blastocyst formation in mice. DNA fragmentation in the sperm interferes with the amount of phospholipase C, decreasing its concentration sufficiently to interfere with fertilization and cleavage. Different mutations and damage will produce varying reductions in the amount of phospholipase C. It's another way that sperm DNA fragmentation may impact IVF and not one currently on anybody's radar. So what is phospholipase C? It's the dominant sperm-specific factor in all the sperm head in all mammals, responsible for triggering the oocyte activation. Where you have a genetic mutation in the gene, you end up with oocyte activation deficiency, or OAD, which has been linked to numerous phospholipase C mutations and abnormalities in patients who experience total fertilization failure, both with IVF and ICSI. In humans, the most dominant region for phospholipase C is the equatorial region, and they produce some figure here of the sperm head, the equatorial region with the staining of phospholipase C and the two overlaid. The function of it, I think I'll do one or two separate videos on phospholipase C because it's really important, and all embryologists should be absolutely aware of this critical enzyme. Basically, as I understand it, as soon as the sperm membranes fuse, one of the first things that happen is phospholipase C, which is found in the sperm head and the equatorial region of the sperm, activate or hydrolyze PIP2 to form IP3, which binds to receptors on the endoplasmic reticulum, which causes a release of calcium. This occurs in waves, and as I understand it, the waves vary in intensity, and you can get variations in the peak and the frequency, which has different outcomes in IVF. These releases of calcium also release the excitosis of cortical granules, causing the zonal reaction and stopping any further sperm membrane fusion, and also the release of the oocyte from meiotic arrest. In mice, in a paper published last year in Human Reproduction, you can demonstrate that if you have mutations in mice with mild or severe damage to the PLC gene, that with mild mutations cause a reduction in the fertilization rate and consequently the rate of blastocyst formation, but a severe mutation basically leaves you fail fertilization. There's quite a lot of different mutations in PLC, and a lot of people have spent work on it. It's not the function of this video, I don't think, but browsers may like to go back and explore those journals. They can demonstrate that the kinetics, the morphokinetics for embryo development in mice is significantly different overall for the mild form of mutation, where that is where you have some phospholipase C, but not very much, and it may even be a slightly altered form. It's not really dramatic, but you can see that there is a growing delay in getting to blastocyst. In humans, the only real change in morphokinetic milestones was the CC2 phase, which is, I understand, the time from 2 cells to 3 cells. And normally it's around about 8 to 12 hours on an ideal KPI. But if you go back and look at the amount of fluorescence in the original sperm sample in humans, the highest amount of fluorescence was found in those samples that led to fertilization, and most of the embryos are in this 8 to 12 hour phase. When embryo development was delayed or accelerated, it was often associated with reduced amount of fluorescence and the density of the fluorescence. So this is a fairly broad linking between a few sperm that were used to initiate fertilization, usually with ICSI, and the sperm in the overall sample. And I think that one of the problems with this sort of work is you're looking at all the sperm in the sample, but it's actually the sperm that you're choosing to do the fertilization, which should really be tested for LPC. But the bottom line is, in humans, you can see a relationship between the amount of fluorescence for PLCz in the semen sample and morphokinetic changes in the embryo. And even making this link even more tenuous, you can suggest that samples with reduced amounts of immunofluorescence, or the proportion of sperm that has got standard fluorescence, is lower in cases where embryo development is delayed or enhanced. This is really talking about the amount of phospholipase getting into the egg and producing a series of calcium oscillations at a certain frequency. And this data does suggest that whether that frequency is delayed or processed going too fast, accelerated, then you have impacts both in the rate to which embryo divides early on, this is the 2 to 4 cell cleavage stage, and you can look at the pregnancy rate and show that the pregnancy rate for embryos which have the normal rate of development, 8 to 12 hours for this step, have higher pregnancy rates. In fact, it's double the pregnancy rate. And so there may well be in cases where you have very low reduced fertilization and lower pregnancy rates. It may be that we're looking at the effects of phospholipase Cs in the original sperm sample in the male. Okay, enough of this background. Let's get to this paper, which is a short paper. And the authors compared two groups, a fertile group that had normal IVF fertilization and an infertile group that had two complete total failed fertilizations with ICSI. So there were no oocytes fertilized with no PN, while the control consisted of men with proven fertility. And the female partners were theoretically all normal. And just looking at the fluorescences, what the authors were observing, you can see that you'd have fluorescence both in the acrosomal region and the equatorial region. And when you put the two together, you can see the equatorial region and the acrosomal region. I make the point here because early on I said that it's thought in humans the primary location for phospholipase C was the equatorial region. But in this paper, the authors are looking more closely at the acrosomal location and showing relationships. So basically, they looked at the proportion of sperm that had fluorescence for phospholipase C in both the control group and the ones with total fertilization failure, and not surprising, they had highly significant fewer sperm that had phospholipase C. There are a couple of points. First is that in the fertile sample, only 65% of sperm had detectable phospholipase C in the sperm head. And secondly, with the total failed fertilization, it was about 18% of sperm that demonstrates phospholipase C. But the real question will be whether the immunofluorescence is picking up phospholipase C, which may not be physiologically functional. And this hasn't really been discussed in the paper, as I couldn't find. And once again, if you look at the intensity, it's not quite as dramatic, but in the fertile groups, there was a fluorescence in the AU units, 60%, 60, and significantly less in the infertile groups, but there's still a fair amount. It's just as a matter of intensity. So you can make a comment that the mean relative total phospholipase C fluorescence level exhibited in sperm from fertile and infertile groups showed that the infertile population, the amount of phospholipase C was significantly diminished compared to the fertile controls. It seems it is a matter both of frequency of the number of sperm with fluorescence and the intensity of the of the phospholipase C, just how much is there. So this may impact the oscillation that the sperm are able to induce in the oocyte after fertilization. And this is the crux of the paper. They then went back and looked at the percentage of sperm demonstrating phospholipase C in the fertile groups and the failed fertilization group. You can see that's the data we showed before, but they can also show that there is in the sample of infertile people with total failed fertilization, there was significantly higher I just need to stop for a second. Sorry about that. You can demonstrate that in the fertile samples, the percentage of sperm showing fluorescence was about 65% compared to 18% with the failed fertilization group. But we go back and look at the degree of sperm decondensation. The normal group had only very low levels of 10-15%, but more than 30% of sperm in the infertile group had damaged decondensation, and likewise with sperm DNA fragmentation. So the comparison of the mean percent fertilization of spermatozoa presenting with phospholipase C's, DNA fragmentation, and chromatin decondensation, there was a significant inverse correlation between the frequency of phospholipase sperm and sperm DNA condensation and fragmentation. And also the authors pointed out that the difference between the fertile and infertile group was not so much in the equatorial located PLC, but in the acrosomal located phospholipase C. And just last year, they wanted to make the point that they could, if they looked at did the correlation between percent of sperm with phospholipase C, DNA fragmentation, or sperm decondensation, they were all significantly related to sperm concentration. They were not related to motility, and only PLC was related to morphology. So this is a group that had total fertilization failure, and you can demonstrate that in that group they had very low levels of PLC, high levels of fragmentation, sperm decondensation errors, but they all otherwise appeared to have a normal sample. So just to summarize this paper, the authors note there is a significant decrease in phospholipase C's and zeta expression levels were observed, especially with those who presented with DNA fragmentation and sperm chromatin decondensation greater than 30%. All the infertile men exhibited overall normal sperm parameters according to WHO guidelines, with significant difference observed in sperm count and morphology, the relationship. However, they presented abnormal DNA fragmentation and chromatin, highlighting the crucial importance of complementary sperm function tests in ensuring successful fertilization and preserving male fertility. And I guess you can make the point here is that if you have a case with a normal semen profile and failed fertilization, one of the tests you should do maybe before the couple return is the phospholipase C staining. And this probably suggests that most clinics should develop the skills or a reference lab to have that done. This data suggests that DNA fragmentation can impact the phospholipase gene in some way. The human phospholipase gene is composed of 15 exons situated on chromosome 12, demonstrating a genetic connection between oocyte activation deficiency, AOD, and PLC, and thereby impacting the function and structure of spermatic protein. That's just what the author said. It's therefore evident, though, that as DNA fragmentation or chromatin decondensation errors increase, the expression of the spermatic protein PLC corresponding will decrease. Oocyte activation failure occurs when the mechanisms required to initiate development after fertilization are damaged, and phospholipase C is a soluble factor recognized as being an essential protein for a successful fertilization while activating several pathways. It precedes nuclear development and DNA repair. But it strikes me that while fertilization failure is a specific consequence of poor or damaged phospholipase C activation, very low levels of the enzyme may have a more subtle impact on fertilization development, and this may have a subtle aspect through DNA fragmentation. So sperm DNA fragmentation may therefore influence in subtle ways embryo development by reducing the concentrations in phospholipase C, the sperm head, and the two features of DNA fragmentation or decondensation and PLC concentration should be seen as related and acting together to limit blastocyst development and the chance of pregnancy. So I think I'd like to stop there, just make a small disclaimer. I hope this information may help you in your fertility treatment. I'm not a medical practitioner, and this information is not medical advice, but it may help clients who are having IVF to know what the doctors are talking about, or maybe help to understand the advice they're receiving. If you like the information I'm presenting, you may love you to subscribe, press like, or make a comment. And finally, once again, this is supported by my company FertAid, which is an online EQA program in IVF, in andrology and embryology. And it does do sperm DNA fragmentation with a hollow sperm. And the embryology courses looks at follicles, oocytes, zygotes, cleavage, blastocysts, in images and time-lapse assessments, embryo raking. You can control as a trainer, as a professional. You can run as a small lab or large multi-lab clinics. It can be used for training or competency or accreditation. I'd like to thank you for your time, and I'll just finish there. Though I'd like to make a note that IVF is a personal journey that involves the action of many staff and many different factors, and it helps to understand all aspects of that journey. I hope this information be of value. If you like the information provided, tell other IVF clients about the service, and please subscribe.